Hello everyone, it's Mr. Wolfe here, and in this video I'm going to be going over the biotechnology of recombinant DNA. So whenever we have recombinant DNA, we're taking DNA from one organism, in this case a human, and we're inserting it into the genome, the DNA of another organism, in this case a bacteria. And the reason why we're doing it in this case is so our bacteria could actually produce a human protein. So why would we do this? Well, let's say that we are trying to treat someone with type 1 diabetes. They're not producing the protein insulin. We could have bacteria make that insulin protein for us. But first we need to make sure that we insert that insulin gene into the bacteria. So this is how the process is going to go down. We're going to have two step ones. The first step one is to isolate. So we'll say, we isolate the gene of interest. In this example, an insulin gene from a human cell. And so here's our human. We're going to kind of zoom in a little bit to their cell. They're going to have a bunch of DNA in their cell, and we're going to extract our gene of interest. So that's the first step one. The second step one is we're going to take our bacteria, and bacteria have these things called plasmids. They're circular, it's essentially just circular DNA. So we're going to take our plasmid out of the cell, or out of the bacteria. So all of this happening right here, that's all happening in a test tube, essentially. So to describe this process, we're going to say our plasmid, and our plasmid is going to be our vector, right? Remember, think of it like a vector. It's like our vehicle. It's our way for inserting the gene of interest into bacteria. So to kind of finish this, the plasmid is taken out of the bacteria. Remember, our vector in this case is our plasmid. All right, so now we have our plasmid, we have our gene of interest. What we need to do is we need to incorporate this gene of interest into the bacteria's DNA. So what we'll do is we'll take a restriction enzyme, that's this guy right here. So this is our restriction enzyme. And if we have the plasmid, some plasmids in a test tube, the gene of interest in the test tube, and we add this restriction enzymes cut at very, very specific sequences. So the restriction enzyme is going to cut once on the bacteria, and then it'll cut twice on our gene of interest. So we'll summarize that right here. Restriction enzymes cut the plasmid and gene of interest. So one thing you'll notice is that when our restriction enzyme cut our enzymes, what we get are these kind of just not free nucleotides, but unpaired nucleotides at the ends. And these are called sticky ends. And notice that we also have sticky ends on our plasmid that was cut. So these sticky ends end up being really important, because what we're going to do is we're going to take our gene of interest and we're going to take our plasmid and we're going to combine the two together to form something like this. And our gene of interest is part of our bacteria. In this case, we could say that we have recombinant DNA. So how did this gene of interest attach to the plasmid? Well, if you notice here, we have a TT, right? It's going to be kind of tough to draw this right here, so I might actually draw it on the outside. TT, and then we have this AA sticky end over here. And then on the bacterial plasmid, we have this AA at the top, and then at the bottom right here, we have a TT. So if you notice, the sticky ends match the sticky ends of the plasmid and the sticky ends of the recombinant DNA. They match up perfectly, and that's because they were cut by the same restriction enzyme. So to summarize this whole process, we'll say that the gene of interest becomes part of the plasmid because the sticky ends match up. So at this point, we have recombinant DNA, but we're not done with our process yet because we want to actually insert this recombinant DNA back into the bacteria. And that's exactly what we're going to end up doing. So our next step is to take this recombinant DNA and put it back into our bacteria. And as I'm drawing this arrow, I realized I forgot a couple other things, which is just to draw kind of an arrow right here, showing that this is going from our processes. I'm also just going to draw my arrows a little bit thicker here, so you can kind of see what are the main steps of the process versus just pointing to things right here. And I'll make these lines a little thicker too. So for step 4, we're going to say that we reinsert the recombinant DNA now back, or just into bacteria. The plasmid's going back into the bacteria. The gene of interest was never in there in the first place. So we reinsert the recombinant DNA into the bacteria. So that's kind of the first step of step 4. Because now that we have the recombinant DNA in the plasmid, what ends up happening, and what's kind of drawn right here, is this looks like an enzyme doing transcription. So the bacteria has the machinery, just like our cells have the machinery, to do protein synthesis. And after we make our mRNA molecule, I'll even label this mRNA. Right, that mRNA goes to our ribosome. And what we're going to end up getting is a protein out of this. So here is our protein. And obviously this protein's not going to be this big, but the reason I'm drawing it this big is because the whole purpose of doing this process of recombining DNA, putting it back into the bacteria, is to make our gene of interest has now made our protein. In this case, our protein is going to be insulin. So to summarize this process, we could say that we reinserted the recombinant DNA back into the bacteria, and now bacteria does protein synthesis to produce the needed protein. All right. And that is recombinant DNA.